Anyway, it's a pleasure to welcome the last speaker of our session here, Tony St. Ledger. I've known Tony since he was a graduate student in Robert Hendrick's lab at University of Pittsburgh. He passed up coming to my lab for a postdoc and instead went to Rachel Caspi's lab at the National Eye Institute. But give him credit, he's done quite well. A beautiful paper out in Immunity, which is probably the most convincing story that I've seen anyway, showing a role for commensals and the microbiome in the eye, albeit in the conjunctiva. Anyway, after Tony managed to get a K99, the so-called kangaroo grant, and he just started his position at the University of Pittsburgh as an assistant professor. So he is one of the young stars in our group. And welcome. Thank you.
Thank you, Eric, for the introduction. I thank the organizers for giving me the opportunity to present some of my work. So today I'm going to give a brief overview of what we published in Immunity. Then I'll talk about some stuff I'm still finishing up at NIH in collaboration with Rachel Caspi. And then I could sort of whet your appetite with some of my stuff I've started in my short time at Pitt so far. So I work with mucosal tissue at the ocular surface, and that's composed of multiple parts like the lacrimal gland, the lacrimal duct, which drains the tears. But my tissue of interest currently is the conjunctiva. And being many ophthalmologists in the field, we know that in the audience, we know that the conjunctiva is a thin mucosal tissue that attaches to the peripheral cornea, goes around, and attaches to the eye. But animal studies and human studies have shown that within this mucosal tissue, there's actually all the ingredients required for a conventional mucosal immune response. There's adaptive and innate lymphocytes, there's M cells to process antigen, there's lymphoid follicles, and we all know that IgA is secreted in the tears and is the main protective antibody of the ocular surface. So if we compare the cells we see in the conjunctiva to a conventional mucosal immune tissue like the intestine, you see that all the cells are there. The M cells, the innate and adaptive cells, you have goblet cells producing mucus. However, it's well known in the intestine that you need commensal bacteria to appropriately tune that immune response to help prevent infectious disease as well as prevent autoimmunity. However, at the ocular surface, the thought was not the case. The ocular surface is conventionally thought to be sterile. And if you're looking at the central cornea, I would agree with you that if you take a swab of the central cornea, you're not going to get any bacteria or very little, if any. Also, because of the constant tear washing, the lysozyme, antimicrobial peptides, and IgA, again, it's not a very hospitable environment for bacteria. However, if you go underneath the eyelid and start taking samples of the conjunctiva and doing advanced PCR analysis, you can see that bacterial signatures are actually starting to form between ethnic groups, geographic groups, as well as people that wear contact lenses or don't wear contact lenses. However, finding DNA evidence, as Katrina showed, as explained, doesn't tell us anything. Are these bacteria alive? Is this DNA that has been there for a while or is just flown there from the environment? So we decided to ask if there are bacteria in the conjunctiva. And we use a mouse model because getting enough human specimens and to do functional analysis was impossible for us. So we started off the complicated way using 16S gene sequencing analysis, and we found that there was a very limited diversity of bacteria at the ocular surface. This confirmed to us that we're probably not getting any sort of contamination from the gut. We had a very limited diversity. And if we look, we see staphylococci, which is known to be there. We see Corynebacteria, which are known to be there, as well as various other commensal and pathogenic bacteria. But again, we found DNA. This doesn't tell us whether these are alive. So we decided to just take bulk conjunctival homogenates and plate them on agar plates. And we found a very interesting candidate. And I could tell you the serendipitous nature of this finding, maybe at the poster session. I don't really have time to explain it now. But what we found was Corynebacterium mastidides actually grew out of the conjunctival tissue. And when we sequenced this bug, interestingly, and connected to human studies, is the isolate was actually very similar to an isolate found in the conjunctiva of a 9-year-old boy that was published many years ago. Further, another case to make it a little more translatable is that this bug is specific to gentamicin, which is a common antibiotic prescribed for ocular surface disease. So it does have a little bit of translatability. But again, we didn't know if this bug had just landed there or just was inoculated by a mouse scratching its eye. So we decided to do fluorescence in situ hybridization to look for Corynebacterium, the genus. And what we found in our mice that were bred in our facility, you start to see that it's actually very abundant in the conjunctival tissue. We see the lighting up, and we actually see filaments start to form in the tissue, suggesting that this bug is actually dug into the tissue and not just flying there or just being introduced there right before the assay. What was, again, serendipitous to my project was that we started to assay mice from different commercial vendors, and what we found was the bug was not present in those. In Jackson Laboratories, we looked at Taconic and Charles River as well. So this gave us a nice model to start to test causality. So the first thing we did to look to see if this bacteria actually mattered was we tossed on some gentamicin onto the ocular surface, a typical regimen of once a day for 6 days. And we collaborated with Mihalis leonakis at NIAID, and we used a Candida albicans infection, so a fungal infection as sort of our infectious model. And we did this mostly because his lab was upstairs and we were very good collaborators already. And what we found in vitro was that if we isolated tears from wild-type mice or wild-type mice that were treated with antibiotics, we see that tears from wild-type antibiotic-treated mice actually were poorer at killing fungus in vitro. But this was an in vitro assay. What happens in vivo? And when we look in vivo, we actually saw a very striking result, which is standard, that if you challenge wild-type mice with Candida albicans, they're naturally resistant. So we see that the cornea after 36 hours remains smooth and glassy and clear. And if we do a PAS stain of the cornea, we see that maybe there's one fungal filament, but no real overt disease. However, if we look at the wild-type antibiotic-treated mice, we see that textures start to form in the cornea, and we start to notice some pretty serious pathology. And you could start to see the fungal filaments actually penetrate through the corneal epithelia down into the stroma. And we used immune-deficient mice as nice controls. You can see that the cornea is essentially destroyed. We confirmed our result using qPCR in collaboration with Mihalis' lab to show that, yes, indeed, if you disrupt the microbiome at the ocular surface, you're going to leave yourself more susceptible to this fungal challenge. But this didn't establish a causal link. So we noticed that if you take the bug away or take all of the bugs away, you see this response. We had to show that we can add this bug back and see a phenotype. So we had a very simple inoculation scheme for inoculating vendor mice with CMAS. So I told you that vendor mice were CMAS negative earlier. So we gently dried the tear film once every 3 days for a total of 3 treatments, and then applied some CMAS to the ocular surface. We then waited up 5 weeks. We've taken out to 8 or 10 weeks, and we looked at CFU by eye swab of the conjunctiva. And what we found was that CMAS was still there in the conjunctival tissue well after our last inoculation. This told us that the bug wasn't just washed away, it stayed there and dug into the tissue. Interestingly, we tried other Corynebacterium species as well as Staph epidermidis, and we saw that doesn't hang around very well. So CMAS, that told us that CMAS is a unique colonizer of the eye and has something special about it that lets it stay around. Interestingly, if we co-house a mouse that's not CMAS positive with a mouse that is CMAS positive, a typical way to sort of get the microbiota mingling, we don't see that the bug transfers to any of these CMAS negative mice. So it's not a horizontal transfer. But if we use this mouse as a breeder and we look at all of her pups, every one of her pups in subsequent generations has CMAS in the ocular tissue. So this supports the notion that this is a resident commensal that is colonizing the tissue and is present. And you can see from our fish staining the confirmation of this. Finally, we challenged mice with fungus as well as bacteria to see if CMAS was imposing a beneficial immunity. And that's exactly what we see. So we look at our fungal genome copies and we see significant reductions in the fungi infecting the tissue whenever CMAST is present. We collaborated with Mihaela Gedhiva at Brigham and Women's, who uses a Pseudomonas aeruginosa infection model, and we again saw a similar reduction in bacterial burden as well as pathology score. And this was actually performed in her laboratory in Boston, so this was nice confirmation for us that our phenotype just isn't because it's NIH. We see that it also crosses institution borders. So this let us establish sort of a causal relationship between Corynebacterium mastidides with infectious ocular disease. So I won't get into the granular details of this interaction now, but what we found was that antigen-presenting cells in the tissue stimulate gamma delta T cells to protect the ocular surface. And my lab currently is very interested in these gamma delta T cells, and I feel like being some clinicians out there, these gamma delta T cells should be pretty important to be looked at, especially in the ocular tissue. Gamma delta T cells are not conventional alpha beta T cells that normally develop after birth. Gamma delta T cells are fetal derived, and they seed barrier tissues like the conjunctiva very early, before birth and after birth. But interestingly, up to 50% of the conjunctival lymphocytes are actually gamma delta T cells in both humans, and I found that in mice. And gamma deltas are linked to infectious disease, wound healing, and autoimmunity at the ocular surface. So the jury's still out whether, I guess, in certain situations they can be beneficial or pathogenic. But what we found when we challenged mice with CMAS, so we inoculated them conventionally, we see that wild-type mice, again, there's no phenotype because this bug is beneficial in the normal steady-state conditions. However, if we inoculate RAG2 deficient mice, so these mice have no T cells, no B cells, they start to develop a keratitis in the ocular tissue. If we look at just gamma delta T cell knockout, so this is a mouse that has no gamma deltas, we see that they also develop a keratitis and have an inability to control bacteria in the ocular tissue. And I think this is interesting because if you look in the literature, Corynebacterium species are commonly linked to eye infections in the elderly. And talking to some of our clinicians at Pitt, they basically push Corynebacterium species off as a standard commensal that, again, they just toss away. They don't link it to the actual disease. However, you can imagine that as your immune system starts to deteriorate over time, and perhaps maybe your gamma delta T cells aren't as effective, maybe that gives Corynebacterium an avenue to stop being a commensal and start being a pathobiont. So we're now asking the question, can we achieve a better understanding of gamma delta T cells functionality to alleviate ocular surface disease? So independently of that, we're also looking at manipulating all of these relationships to see how we can swing a disease state. And before I left NIH, we started collaborating with Warren Strober and Ivan Fuss, looking at these IL-1 beta and gamma delta T cell interaction. And instead of using an infectious model, we're using an autoinflammatory model, specifically Mucosal syndrome, which is pathology associated with the overproduction of IL-1 beta. It's caused by a genetic mutation in the NLRP3 inflammasome, and primary manifestations of this disease in humans are in the skin, joints, and especially the ocular mucosa that you can see here. So the effect that Warren and Ivan showed was primarily mediated through APCs and Th17 cells. And there are effective therapies like anakinra, which is an IL-1 beta blockade, but we still don't quite know why ocular disease actually manifests. So we ask, can ocular surface disease be due to an aberrant response to commensals? So this is some background from Warren and Ivan's original paper linking commensal bacteria in the skin to spontaneous dermatitis in a Mucosal model, mouse model of disease. So these mice have a homologous mutation that's seen in humans. And you can see that the Mucosal mice develop spontaneous inflammation that's then alleviated whenever you treat with systemic antibiotics. So we came up with the hypothesis that this is probably a systemic response. So we induced DSS colitis in these mice, and we see that colitis is actually more severe in Mucosal syndrome mice, but we saw no eye phenotype. So this told us that it's probably not some dissemination of gut microbes causing ocular surface disease. So we asked the question, can commensals at the ocular surface actually be causing this disease? And we first looked at a spontaneous model of the mouse strain. So you can see Mucosal, wild type compared to Mucosal. And these Mucosal mice developed the spontaneous dermatitis. And you can see from the eye-draining lymph nodes, their lymph nodes are huge, gigantic, some of the biggest I've ever seen in a mouse. If we look at the ocular tissue as well as the eye-draining lymph nodes at gamma delta T cells, we see that there's a lot of IL-17 produced by these cells, which we're hypothesizing is pathogenic in this model. Similarly, if we look at neutrophils as a function of as a measurement of IL-17 functionality, we see that neutrophils are significantly increased. Now, this is a spontaneous model. We have no idea if CMAST is actually present. But the problem is we could never actually attribute causality because these mice develop dermatitis so frequently. So if you back cross this mouse strain to the white BALB/C background, they actually don't develop spontaneous inflammation. So this allowed us to actually test causal relationship. So if we inoculate wild-type mice and Mucosal mice with Corynebacterium mastitis, you can see that a fraction of them start to develop this really severe conjunctival phenotype. And we can actually see this phenotype occur 3 weeks after that final inoculation if we compare neutrophil numbers in the conjunctiva. So without CMAST, there's really no difference. However, if you add CMAST, we see this slight increase in neutrophils that's usually non-pathogenic, but we see a significant increase in neutrophils over the wild-type control. This suggested to us that CMAST can cause this phenotype. As confirmation, we looked at gamma delta T cell activation. We see that they're significantly increased in the draining lymph nodes, and we also see an increase in IL-17, again supporting our thought that these gamma delta T cells are actually very overactive. So we next tried to get a little bit of a mechanistic detail, what's actually going on here. So we performed a simple mixing experiment. So we got antigen-presenting cells from wild-type or Mucosal mice, and we co-incubated them with CMAST and gamma delta T cells from wild-type or Mucosal mice. And the conclusions from this are a little bit complicated, so I'll walk you through it. But we're seeing a very interesting phenotype in these mice. So if you compare wild-type cells that are stimulated with wild-type DCs, or antigen-presenting cells, or Mucosal antigen-presenting cells, we see a huge increase in IL-17 production, suggesting that antigen-presenting cells or gamma delta T cell extrinsic factors are in play. So this is what the literature has stated. However, if we compare wild-type gamma delta T cells and Mucosal's gamma delta T cells stimulated by the same antigen-presenting cell, we're still seeing about 2 to 3-fold more IL-17 produced, suggesting that there's a gamma delta T cell intrinsic. So there's an inflammasome property within these gamma delta T cells that could perpetuate the problem. And you can see from the flow cytometry, comparing these frequencies, that the flow is very convincing in my eyes. So we actually look for inflammasome components in these gamma delta T cells to see if this could be a factor. So when we perform mRNA analysis of components of the inflammasome, caspase-1 and ASC, we see that Mucosal's gamma delta T cells actually have more inflammasome components compared to the wild-type control. If we look at pro-caspase-1, so it's an inactive form of caspase-1, we see that Mucosal's gamma deltas always have more of the component compared to the wild-type control. But functionally, we did a functional assay using this FLICA, which measures active caspase-1 activity. We see that the Mucosal's gamma delta T cells has a higher MFI, so it's cleaving this reagent much more effectively than the wild-type control. And this is similar to some studies done at NIH looking at CD4 inflammasome activity as well. And you can see the quantification here. So with this part, we are trying to say that overactive inflammasome activity could increase susceptibility to pathology associated with IL-17 gamma delta T cells. And there might be a meaningful intrinsic inflammasome role in the gamma delta T cell compartment. And in certain situations, CMAST, which is usually non-pathogenic, can actually trigger the inflammasome and become a pathobiont in Mucosal syndrome. So in the last few minutes, I'd like to go over the animal model of HSV, which I'm working with at Pitt. And I'll just very quickly go over it, where HSV, it can cause blindness in humans and cause blinding keratitis. And when we infect, it starts to replicate in the cornea epithelia, gain access to axons innervating the cornea. It can then travel through retrograde axonal transport back to the trigeminal ganglia where it goes latent. And periodically it'll reactivate, get delivered back to the ocular surface, causing disease. So there's two places of viral control. At the surface before infection, you can push it away before it has a chance to infect, or you can control it back in the trigeminal ganglia. Currently, I'm focused with the acute phase, so within a few days, trying to eliminate virus from the cornea early. And the reason why this is kind of important is because neonatal HSV is actually a very severe disease. So 45% of neonatal HSV is associated with skin, eye, or mouth disease, so basically cold sores in the skin or mouth or eye. However, 30% of them have CNS involvement and 25% actually have disseminated disease. Both of these could end up in death or lasting damage. So we're trying to figure out how to, especially in neonates, reduce disease. And some very, very preliminary data we see here, we're trying to link CMAST to viral protection. So our experimental setup is that we inoculate mice with CMAST as normal, we infect with HSV, and look very early after infection to see if there's any differences. And the differences are striking. So if we look at 2 days post-infection, so basically the virus is replicating in the corneal epithelium, we do a fluorescein stain, you can see that there's very nice green suggesting that there's epithelial damage. However, in mice that have CMAST, you don't see such an intense stain. You maybe see some at the periphery, but there's really not anything in the cornea. That doesn't mean these mice are not infected, because at 6 days post-infection, you start to see some ocular swelling. However, if we look in the cornea for monocytes, we see that there's a significant reduction in monocytes whenever mice are colonized with CMAST compared to the HSV-only controls. Then, if we look even further and we start to see whenever the acute phase of the response starts to calm down, you could see that mice simply infected with HSV-1 actually have a very severe skin disease phenotype, which is alleviated very quickly in HSV plus CMAST mice. So this suggests to us that somehow CMAST is imposing some sort of benefit at the ocular surface, not only in fungal and bacterial infections, but also viral infections. So this is an area of active research that we're performing right now in the lab. So with that, I can thank the people in my lab. So we have a few postdocs now, and I inherited actually Bob Hendricks' people, who has an impending retirement in July. We also have Flowcore and our collaborators, Kip Kingington and Ben Treat, as well as my funding. So then I could take any questions. So in regards to neutrophils producing IL-1 beta, we haven't looked at those. So CMAST in particular, a lot of the sequencing data haven't gone that deep. So I think between 50 and 60% of humans have Corynebacterium, the genus, in the ocular tissue. Whether that's mastidides, we're not sure. But again, our pathologists at Pitt have said that from corneal scrapings, whenever people have disease, they always find there's a lot of Corynebacteria in there, but they push it off as I think technically it's a non-diphtheroid Corynebacteria or something, which they say is a normal commensal. So, but it could be causing a lot of that pathology. So in the skin specifically, so Yasmin Belkade has showed that Corynebacterium in the skin is usually Corynebacterium accumbens, not mastidides. They haven't found it. In regards to the mice, I think actually Corynebacterium is probably more prevalent in universities as opposed to vendors, and I think we need to start taking that into consideration, that vendor mice are very clean. So we had mice shipped from WashU and St. Louis to do some other experiments with collaborators, and they had it, and they came into our animal facility, and mice in our animal facility had it. So potential criticism was that, oh, this is just unique to NIH. I think the Jackson phenotype is unique to vendors, is how I would say it, not necessarily the other way around. So we do know that, so there are CD8s and CD4s that get stimulated by it, and they don't traffic to the con specifically, because we haven't seen a real boosted alpha beta T cell response there. We haven't looked at whether we're getting an expansion of HSV specific cells very early. I think it's very possible that that the bacteria almost acts as an adjuvant at the point of infection. Almost, you have this smoldering inflammation that whenever you get this bug, that the immune system can go into overdrive much more quickly than if it's a sterile environment. And in regards to your comment about the bacteria, so what's interesting, whenever we have our mice start to develop a lot of skin disease, the diversity of bacteria goes through the roof. So in asymptomatic mice or mice that aren't infected with HSV, we see only Corynebacteria there whenever we colonize them. But when we infect them with HSV, you'll get a bunch of other bugs that pop up. And I think it's probably because you're opening up a skin niche that Corynebacterium can overcome.
Right.
Yeah.
Great. Very nice talk. Thank you.
Thank you.